today we're going to be talking about our first of its kind AI-powered search tool for reagent selection and experimental design. My name is Poppy Nathan, and I'm a scientific marketing specialist here at Protein Tech. So just in case you are unfamiliar with Protein Tech, we are a leading life science reagent manufacturer, and we manufacture proteins such as antibodies and immunoassays against the human proteome. We currently have antibodies against over 14,000 human targets, with the aim of producing an antibody against every single protein in the human proteome. As well as this, we have immunoassay kits such as IHC kits, ELISA kits, and cell separation kits. We have our humankind range of recombinant proteins. So all of our cytokines and growth factors are produced in HEC293 cells. So they have all of the native folding and post-translational modifications that you'd expect to see in human proteins. And as well as this, all of our humankind proteins can be made at a GMP grade. So if you are doing any work that you can see becoming clinical, where you're going to need some GMP grade products, you'll be able to use the exact same products without needing to revalidate. In 2020 then, we acquired Chromotech, so our portfolio now encompasses a range of nanobody-based reagents. And nanobody-based reagents are really exciting because they are about 10 times smaller than traditional antibodies, so they're really good for things like protein purification or very high-resolution imaging, as well as cell and tissue penetration. And finally then, we have our subsidiary Protein Tech Genomics, where we have a range of DNA-conjugated antibodies for use for proteome investigations alongside your single-cell RNA sequencing. So we are a company of scientists for scientists. So all of our award-winning products are manufactured in-house, which means we can keep the quality control along all steps of the manufacturing process. We also understand the importance of validation data. So we have the highest level of product validation, and we share all of that data with our customers. We have PhD level technical support all throughout the company, and particularly to help you with any immunoassay questions or struggles you might be having. And finally, we are a global company, so we have 7 inventory sites in manufacturing and research across 3 continents, so we can do global next-day delivery. And we always just like to highlight that we define our success as a company throughout your success. We currently have over 350,000 citations globally, which puts us at the top 4 cited antibody companies. And if you are familiar with some of our competitors, you'll understand why we're so proud of that figure. So coming on to today's talk then, as I mentioned, we're going to be talking about some of our industry-leading AI-powered search tools. We're going to start off with our Able AI, and then I'm going to introduce you guys to the 3D epitope viewer. If you do have any questions along the way, please feel free to pop them in the chat or put them in the Q&A box, and we'll try and come on to those at the end. So I just wanted to start off then and highlight why reagent selection can be so difficult and why this is something that we really want to help optimize for our customers. So first of all, there is an overwhelming number of products to choose from currently on the market. There's lots and lots of antibodies available against the same antigens, even from the same company. And as well as this, antibody performance is really dependent on many different variables. So as I'm sure you guys are aware, antibodies behave very differently depending in different applications, different sample types, different sample preparation methods, and even how your machine is set up in the lab that you're using, as well, of course, between lots of variability that you are going to see between different batches of your polyclonal antibodies. As well as this, there's also a distinct lack of protocol standardization and product data transparency. Because we understand how difficult reagent selection can be, as I say, we're really trying to help optimize this process for our users and ultimately save you guys time and money and let your focus be on the work that you're doing when you're in the lab. So our Able AI, as I mentioned, it's an AI-powered search tool on our website, which helps you find tailored product recommendations for research areas and applications. As well as this, Able can help offer step-by-step experimental guidance, which saves you guys your valuable planning time, reducing trial and error. And also one thing Able can do is it can understand and respond in multiple languages to help democratize global research and really make it accessible for everyone. So you can find Able on our homepage. If you see, we just have a little AI-powered search button next to our main search function. And if you just watch this video, then you can see when you click it, you get a pop-up. And in Able, you can ask any question that you want to help you with your either, as I say, experimental design or for your reagent selection. So in this case, we've asked Able to find us an antibody against CD4 for flow cytometry. And obviously CD4 is a very, very well-established flow cytometry marker. So there are many to choose from, both within our portfolio and also on the market. So once it's had time to think, and you'll be able to always see the thought process that Able shares. So you'll be able to go through and check that everything it's thought about, you're happy with. You'll be able to see that, if I just pause that, you'll be able to see all of the different antibodies that are recommended for you, all of the different preform panels. You can also find on here any validation data, whether or not they're conjugated, unconjugated, and how we recommend using them. As well as this, you can also find Able on its own website at ptglab.com/Able, like in the bottom right, and you can use it in the exact same way you'd use other large language models. But I just wanted to take you through a couple of different examples, really, to help you understand how you can incorporate this into your experimental workflows and how you can really, as I say, optimize this every step along your experimental journey. So starting off at your planning stage then, you can use Able to even help design your experiment right before you've even decided on the reagents that you're going to need. So in this example, we asked Able to help design us a T-cell panel for use on a specific cytometer on the Cytoflex. And Able was able to produce a table of 7 different T-cell markers designed specifically for the Cytoflex. So it gives you the conjugates, the laser excitations, the purpose, the marker that they're looking for. In this case, it's assumed that we wanted to look for human PBMCs because we didn't give it any other information. But again, you can be more specific with that, specific to the sample type that you have. It's got all the catalog numbers there, so you can easily find them directly on the website. But some other examples of things that you could put in to help Able come up with your experimental designing process. So as I mentioned, you could design a flow cytometry panel. You could ask it to help you name the controls that you're going to need, for example, in your Western blot. You could ask to help you put together a recommended order of staining for intracellular and extracellular markers. So you really can be using Able before, as I say, before you've even chosen the antibodies that you're using, before you've even decided if you're buying protein tech antibodies, you could go to Able really from the beginning of your experimental journey. So once you've decided the reagents that you do want, as I say, to save time, trawling through a search function, you can really use Able to optimize, as I say, your tailored reagent selection. So in this case, in this example, we asked Able, do you have an antibody against 1433 gamma that won't cross-react with other isoforms? In this case, Able was able to identify 2 potential 1433 gamma antibodies, a polyclonal and a monoclonal, and it's been able to highlight the precise validation data for you to be able to identify that yourselves. But again, other things you could ask to help you find specific reagents include things like, I need a highly validated antibody against one MMP protein that won't cross-react with another. You could ask the difference between IL-6 ELISA kits. We have several available on our website, so you might at first glance not know exactly what the difference is, and without having to go through to the product data sheets and the product pages, you could just ask Able to be able to differentiate that for you. And finally, you can even add other reagents that you may be currently using or ones that you're comparing it to, and you can ask if we have an alternative for that available on our website. I mentioned earlier, but we're really proud of the fact that Able can respond and understand in multiple languages to, again, help us democratize our global research. So I've just kindly used one of our inside sales team members here to help me generate some questions in both Italian and Spanish, just to show you that whatever language you will put into Able, they'll be able to spit out for you. So you don't need to select this, just whatever comes most naturally to you as you're typing. So on the left-hand side, we've got a question in Italian asking whether or not our GFP trap will also bring down RFP. And on the right-hand side there, we've got a question in Spanish asking to share some publication data about our monoclonal antibody of GAP-DH. So again, this isn't just European languages either. We have a very big Chinese R&D team who utilize this and have helped put this together as well. So whatever it is how you want to communicate with Able, we have the accessibility to be able to answer that for you. And as I've mentioned, validation is something that we're really proud of at our company. So you can use Able to help pull together all of our in-house validation data and all of the published validation data for all of our products. So again, in this scenario, we have just asked Able to suggest a primary and secondary antibody that have validated for use together to look for TDP43 in a Western blot. And again, it's been able to highlight for us a primary and a secondary antibody that have been validated and used together. If you scroll a bit further down from this as well, it linked out to the citations showing what in the different publications of which these have both been used together and what they're used for. But again, not just this, you can ask Able anything, such as, do you have an IL-6 ELISA kit that's been validated in this particular sample type? Do you have any extracellular vesicle markers validated in sheep? Or has this specific product, if you've got a particular product query, you can say, has this specific product been validated in Drosophila, for example? And Able is able to bring all of this for you together to, again, to save you all of that time and save your resources to keep your mind focused on the experiments in the lab. So something that is really important in antibody selection is we always suggest that you use an antibody that has been validated specific for the application that you want to use it in. And this is because different antibodies will work differently in different applications. For example, in some applications, such as flow cytometry or immunofluorescence, you've got a native structure of your protein. So your protein will be present in its tertiary or its quaternary structure. But once you're performing something like a Western blot, when you're denaturing and linearizing your protein's now going to be in its fully denatured primary structure. And depending on the epitope that your antibody binds, it's not necessarily going to be applicable for both of these applications. However, obviously, sometimes it would be better if we could buy one antibody instead of multiple. So in this case, what we've asked Able is, do we have any 1433 antibodies that will work for Western blot and flow cytometry? So in this case, we're asking for an antibody that can work in both the denatured and a native state. And again, it highlights to us all the things that we have. If, for example, we didn't have an antibody available for this, Able would tell you that as well. So it's not only just telling you whether or not it's available, but if you can look down at the bottom there and the key details, it's able to share with you some specific recommendations for these. So it's highlighted the issue that in this case, 1433 proteins are intracellular. So standard flow cytometry without permeabilization is not going to work, that you're going to have to perform that blocking and fix permeabilization step. It's also giving you a recommended starting dilution for the use in each of these applications. So Able's aware that using from the product information that we've put into Able, it can tell that, yes, we do have this antibody, but you are going to have to take into account some considerations to optimize it for each of these different applications. You can also ask example prompts for different research areas, as I said. So in this case, for extracellular vesicle research or antibodies against synaptic proteins, or you could ask, for example, which growth factors should I use to culture my cerebral organoids? So Able really is here to, again, help streamline this process for you and make it a much easier decision-making process. So just to summarize then quickly, Able, as I mentioned, it's an AI powered search feature, which provides tailored product recommendations based on your unique project needs. It delivers relevant background and scientific insight to help you make those decisions. And finally, it strongly builds on the epitope viewer data to help provide deeper experimental insights through validation, citation, and other resources. So I've mentioned our epitope viewer a couple of times now, and I've spoke about the importance of knowing the antibody epitopes. But I just want to spend a bit of time going over this in case you aren't familiar with our other AI tool that we have out, which is our 3D epitope viewer. So when we refer to an epitope in antibody biology, we're referring to the specific binding region of the antigen to which the antibody binds. This is a really, really important piece of information because the epitope does also influence experimental design. Where the antibody is binding will influence things such as whether or not you need to fix and permeabilize, whether or not the multiple antibodies can work together, or whether there's going to be steric hindrance, whether or not as I mentioned, your antibody is going to be able to recognize native proteins or in the linearized version only, whether or not it will target specific isoforms, and finally, whether or not there's any cross-reactivity with other species. So it's really important to be taking this information into consideration when you're choosing an antibody. Despite the importance of this, the epitopes for most antibodies on the market is still unknown. But at Protein Tech, we have launched a brand new antibody validation initiative to help map all of our antibody epitopes. And importantly, we're sharing that information with our customers. So currently, over 2,500 targets have been epitope mapped using a mixture of structural, computational, and fragment-based approaches. And we're sharing this information with you guys via our online 3D epitope viewer. So this is a very user-friendly website tool, just completely accessible again on our homepage, where you can view your proteins of interest in a 3D model, and you can rotate and zoom in on the model to find exactly whereabouts our antibodies bind when we've got multiple antibodies available for the same protein. So in this case on the screen, you can see our TDP43 protein has lots of different potential antibodies available, and it will highlight the exact epitope binding site on your protein. So as well as this, we have our immunogen selector function. So if there's a specific region or domain of the amino acid sequence that you want your antibody to target, you can highlight that sequence within the immunogen selector function, and the relevant antibodies that we have will be available for you. So I just want to highlight quickly then why epitope data is important. So as I mentioned earlier, there's lots of different reasons why you need to take this into account when you're doing your experimental design. Things like your domain location accessibility, isoform and conformational specificity, and cross-assay functionality. So again, that's whether or not it will work in different applications. Cross-reactivity between different species. It's also very important when you're coming on to disease research. So there'll be some disease-specific epitopes. There'll also be different regulation of post-translational modifications in these disease-specific epitopes. And if you know exactly where your antibody binds, you'll be able to know whether or not you're targeting the actual bits that are going to be regulated in these diseases. It's also really important for therapeutic potential. As you can imagine, any therapeutic monoclonal antibodies are going to need to be targeted very efficiently to get that high efficacy and to also reduce any off-target effects. And finally, I just want to spend one more slide talking about this matched antibody pair compatibility that I mentioned before. So for certain applications, it's important to have two complementary antibodies that recognize different parts of the antigen. And this is for many binding assays for things like sandwich ELISAs, FRET, LFT, multiplex IF, or bead-based arrays. And currently, due to the fact that we have epitope mapped several of our recombinant proteins now, we've been able to use this data to highlight potential pairs that should work, and then we've experimentally tested those. And by doing this, we've managed to significantly increase the output of our matched antibody pairs. So we currently have the largest portfolio of matched antibody pairs. We have over pairs against over 3,600 human targets. And for these, we often have multiple pairing combinations. So being able to understand, as I mentioned, that epitope biology can really enhance experimental design and increase your reagent relevance when it comes to your specific project. So if you do want to have a look at your favorite protein or the protein that you're studying, you can just scan that QR code there and you'll be able to go to our website, have a look at your protein of interest and see whether or not we've got any antibodies that bind and whereabouts they do bind. But just to bring everything I've mentioned together then, we, as a company, as I mentioned, we really, really believe the importance of data validation and data transparency. And we're the only company out there at the minute that is driving research progression in this way. We're the only reagent manufacturer that has this AI-powered search tool, and we're the only antibody provider who is actively epitope mapping and sharing that data, as I said, with our customers. But we believe that sequence-defined reagents can give a confidence in reproducibility and flexibility in custom options. So we're really increasing the reproducibility within this field. We're sharing epitope and immunogen level insights to help inform reagent selection, which again, just helps improve that assay reproducibility. And finally, Able AI can minimize guesswork and globally democratizes reagent information, which again, just saves time and costs for you guys, the end users. So I hope you found that useful, and I hope you do use our Able AI search function and our epitope viewer. If you want to learn more about the other support that we offer, please do check out our video hub on our website, or we have free online courses on our learning portal, which you can find at learn.ptglab.com. And with that, I just want to say thank you for listening. I hope, as I said, you found it useful. I will stop sharing my screen and just see if we have any questions. I can see a couple in the chat already. Oh, sorry, I have just momentarily lost the Q&A box. Here we go. Okay. So somebody has asked, are the recommendations for the experimental planning based on publications, chemical, physical characteristics of the antibodies, or both? That is a really good question. So the recommendations that are coming out of Able come from everything that we have on our website and what we've trained our AI model on. So any validation data that we have regarding the characteristics of the antibodies, we've put that information into Able, so we'll refer back to that. But it will also come down to any publications that, again, that we've trained the model on. So all of the citations and publications that you can find in our on our website or that are related to our products, all of that information will be taken into consideration when Able is giving you a recommendation. Somebody has asked, can this tool suggest IHC protocol optimization steps based on tissue type? Yes. So again, Able will has been trained on a whole host of different training metrics, a lot of which is already on our website. But if you want more information about IHC protocol optimization, there's an awful lot available on our website in the support tab. So we have an IHC protocol video, we have an IHC technical support guide. And again, that's got lots of protocol optimization steps. Able theoretically should have all of that information in it as well. So if you've got a more specific question, Able will be able to help there. Definitely, please give it a go and see if it can help you. As always, if there's anything that Able hasn't been able to answer or you need more support on, please do get in touch with our technical support team or our live chat, and we'll always be able to give you more information that we haven't been able to cover through Able. Someone has asked, they want to know if a specific antibody is knockout or knockdown validated. Would Able be able to give you the publications? Yes, absolutely. If we've got publications or internal validation on knockout, knockdown samples, then yeah, absolutely, Able will be able to bring that information and share it with you directly. Can Able AI tell me which antibodies I can multiplex together? Yep, so Able will definitely be able to give a suggestion. It can always help you come up with experimental planning. So it will, for example, probably to make that question a bit more tailored, you might want to mention obviously the experiment that you're using, what your outcomes are, the targets that you want to highlight. If, for example, it's in flow cytometry, then you'll want to let it know what your parameters are in terms of your experimental setup. For all of this, Able is more than able to bring all this information together and again share it with you. So this is a good question. Would Able AI be able to skim through publications for non-protein tech products such as primers? Unfortunately not at the minute. Able has been entirely trained on our product portfolio. So currently that is its limitations and it won't be able to give any advice outside of that. So if you are looking for primer information, then that's one thing that Able is not going to be able to help you with, I'm afraid. And amazing, we've got a few more questions in the chat as well. So is the AI model helpful for adjusting compensations for flow cytometry? Yeah, absolutely, it should be. So on our website as well, we now have, as well as the FluoroFinder spectral viewer, we also have an easy panel panel designer. So again, it's all under our support tab. You'll be able to find it there. And that's also a really good place to start. So for example, you could use the easy panel builder that we have to come up with a flow cytometry model. And then if you're having any issues with that, or you want further information, or you want to be able to ask a direct question, that's where you can then go to Able and see what Able can provide for you. And again, as always, if Able hasn't been able to help you, or there's more specific question that you've got regarding, let's say, a technical support issue, that's when you want to go to our technical support team. Is Able capable to help with antigen retrieval protocols? Yeah, great question. Again, because Able has been trained on the information that we have got on our website, as well as what we've provided, there is information on antigen retrieval protocols. What Able will probably be able to help with most is if you are using one of our antibodies. Obviously, antigen retrieval does vary depending on different antibodies, different setups. It's not always necessary. So if you're using one of our products and you want to find out the specific information on that, Able will be able to provide either the product data sheet or be able to interpret the information off of that and provide it for you directly. Do we have, oh, here we go, one more. Is Able suggesting only specific antibodies or combinations with anti-tag and specific antibodies? Yep, so if we have got some anti-tag antibodies available and you are looking specifically for that, then yeah, make sure that you put that in the question and Able will be able to provide that. If you just ask specifically about the target, it will probably assume you just want the antibody directly against the target. But if you ask clearly that you're looking for an anti-tag, we have other products available as well, not just antibodies against tagged proteins, and Able will be able to help bring together all of the product data with the validation data, citation data, as well as what's in the product recommendations. And hopefully it'll be able to provide you with all of that information. Somebody has asked whether or not you can use an AI-generated summary for a publication. So Able won't be able to generate that for you. So there's no issue there in terms of you using it because it won't be able to be done. And oh, is that one more? Is Able useful for designing spectral surface and intracellular panels that are larger? So it is a good question. And yeah, absolutely, it will be able to help you do that. As always, Able is a tool here to help you guys make these decisions. Able cannot make the whole decision for you. There'll always be things to consider, particularly in terms of a big spectral panel. You know, it'll be really important for you to understand the intricacies of the experimental setup of your cytometer. So in this case, although Able will be able to point you in the right direction, you will probably need to spend more time using other of our tools available, as I previously mentioned, the Flora Finder and the Panel Builder. And then once you've had a greater understanding of the different targets that you want, Able will then be able to, as I say, point you in the right direction of the products that we have to help you with this. Can the 3D epitope viewer give predictions of unknown epitopes? So no, our epitope information is all validated experimentally. The AI use for the epitope viewer is only in terms of the 3D protein modeling. We're using AlphaFold to help predict putting the model together alongside experimental data, but all of the epitope information that we have available, as I say, is experimentally resolved. And maybe just a couple more questions then, but it's really great to see you guys are so interested to learn more about Able. And please do try using Able, obviously, yourselves. And let us know whether or not it's managed to answer your question. But finally, someone has asked, do you include a diverse range of fluorophores that Able can use? So we have a large in-house portfolio of fluorophores. They're called our correlite dyes. So these are very spectrally equivalent to Alexa fluors. So yeah, as when you're searching for our reagents, you'll be able to see all of our antibodies, whether or not they become unconjugated, conjugated, the fluorophores that they are conjugated to, and all of that information, again, Able can bring together and share that all with you. And okay, one more question just popped into the chat. If I use an antibody from another manufacturer, can the 3D epitope use that information? Again, unfortunately not. So our epitope viewer is from experimentally mapped epitopes of our own in-house antibodies. So we won't be able, it's not like an AI prediction tool. The epitope viewer, as I say, has definitely been experimentally mapped. So we wouldn't be able to share it to tell you any information about another competitor's antibody epitope. What we could do, however, is if you ask Able, if you put in the catalog number or the product from the other company and you want to know whether or not we have an equivalent that has been mapped, Able will be able to share that data with you. So thank you everybody for all of your questions. If you do have any other questions, then you can get in touch with me directly. My email was on the slides that I'll send out, or you can get in touch with, as I say, with just europe@ptglab.com. And I know that there was a raffle today for everyone who attended. So as soon as I end the webinar, we will have a look through the attendees. We'll do a live wheel spin probably to choose the winner, and then we will email out whoever that is the lucky attendee. So please do keep an eye on your emails. Make sure you keep an eye on your spam emails as well. You'll receive all of this information within the next 5 working days. You'll receive a recording and a certificate of attendance and link to the slides. As well, of course, we will share the raffle prizes with the winners. So I hope you all have a wonderful rest of your day, and I hope you all found this very useful. Thank you everyone for attending. And with that, I will bring the webinar to a close.